Hello, everyone. So today we have Miguel Sanz-Novo. Miguel completed his bachelor and master studies in chemistry at the University of Valladolid, where he graduated top of his class and received the extraordinary awards for both degrees. In 2018, he obtained the National FPU Fellowship and completed his PhD in chemistry after a research stay at the Max Planck Institute for Radioastronomy in Bonn, in Germany. His research focused on the interstellar molecules and biomolecules, combining high-resolution rotational spectroscopy with quantum chemical computations, receiving his third extraordinary award for the best doctoral thesis in chemistry from the University of Valladolid in 2022. He then joined CAB, first as a Margarita Fellow and later as a Juan de la Cierva Fellow, where he investigates molecular complexity in the interstellar medium and its implication for the origin of life. Miguel has coordinated several research works with the international institutions, such the Max Planck Institute for Radioastronomy and the Max Planck Institute for Extraterrestrial Physics in Germany, MIT and NRO in the United States, the University of Cologne in Germany, the University of Chemical Technology in Czech Republic, among others. He is also a regular visitor of various world-class laboratories around the globe, including the Center for Astrochemical Study in Garching, the McCarthy Laboratory at the Award and Smithsonian Centers for Astrophysics, and most recently, the Star and Planet Formation Laboratory at Riken in Tokyo, Japan. Despite being early in his career, he has received several relevant awards, such the ESEVA Best Doctoral Thesis Award in 2023, the John Eugene Memorial Award in the same year, the award for the best article by a young researcher in atomic and molecular chemistry and physics by the Spanish Royal Society of Physics, as well as that of chemistry and the group of young physics into 2023. And very recently, the Miller Prize from the ESMS conference this year. Since 2022, he also holds the ANEC accreditation as Professor Ayudante Doctor and Professor Contratado Doctor. And today he's going to talk about the work that he has been doing here in our group, titled A Journey Toward the Limits of Interstellar Chemical Complexity, a synergetic laboratory, theoretical, and observational study. Go ahead.
Thanks a lot, Laura, for the introduction. And also, I'm quite grateful for being here after almost 3 years already at CAP. And well, today I'll try to take you on a journey toward the limits of chemical complexity. And as you will see, it will be a synergetic work, a synergetic journey. So astrochemistry can be defined as the study of molecules in space. In the last couple of years, we have seen a dramatic surge of new discoveries with more than 340 molecules detected as of last month. You can see, for instance, the database that is run by Holger, the CDMS, at the CDMS webpage. And interestingly, about 1/3 of the total known census has been discovered only during the last 5 years, primarily towards 2 regions. So ranking first, we find the dark molecular cloud, TMC1. I wanted to highlight the works that both the Gotham collaboration is doing, led by Brett McGuire, with the detection of these extremely large molecules, this, for instance, this 7-ring PAH cyanocoronine, and also the work that Pepe Cernicharo is doing also toward TMC1 with the Quixote line survey. And this group also in Madrid has detected over 90 molecules, so a lot of new detections. And ranking second, we find the Galactic Center molecular cloud G plus 0.693. This is a mosaic view of the center of our galaxy taken with the MIRCAT interferometer. So this particular cloud is located here within the Sagittarius giant molecular cloud complex. This cloud exhibits quite peculiar, I would say, physical and chemical properties, such as intermediate hydrogen volume densities. The gas and the dust are thermally decoupled. And also this cloud shows a very rich chemistry dominated by the action of large-scale, low-velocity shocks, which is essentially grant us access to the chemistry and indirect access to the chemistry that is occurring on the surface of dust grains. And all this encourage Victor Rivilla and Izaskun Jimenez Serra here at CAP to conduct an unbiased, sensitive, broadband molecular line survey using different astronomical facilities toward this particular cloud. And in a first generation of works between 2018 and 2022, this led to the discovery of nearly a dozen new molecules in the ISM, including some of highly prebiotic relevance, such as ethanolamine, which can be seen as the simplest phospholipid head group, and also perhaps M-propanol, which can be considered as a precursor of fatty alcohols in the ISM. And in the last couple of years, we've performed new observing runs, yielding to what we are now calling a new ultra-deep spectral survey of this particular cloud, using, where we perform new JEVES 40-meter observations, leading to a final noise level, superb sensitivity below the millikelvin across the whole Q1, and also new IRAM 30-meter observations, leading to this sort of, again, superb sensitivity at 3 and 2 millimeters, respectively. And with this amazing data, overall, in our group, we have reported the detection of nearly 25 new interstellar molecules in the last 6 years, including species with presenting the 6 key elements for life, so hydrogen, carbon, oxygen, nitrogen, sulfur, and phosphorus, plus some additional refractory molecules. And probably some of my all-time favorites that will also be part of today's talk are the detection of carbonic acid, which is the first molecule detected in the ISM containing more than 2 oxygen atoms in its structure. Also, we reported the detection of glycolamide, which is the first glycine isomer that has been found in the ISM. And then in the second part of the talk, I'll present several sulfur-bearing molecules, because sulfur chemistry is also now a quite hot topic in astrochemistry, such as dimethyl sulfide, protonated carbonyl sulfide, and thionylamide, HNSO. So what is clear is that we are now living a completely golden age of astrochemistry of this particular field. And our goals at CAP is to decode, to decipher what is the connection between the interstellar inventory that we find in the ISM and the chemical content that has been discovered in the minor bodies of our solar system, such as asteroids, comets, and meteorites, which includes a wide variety of key prebiotic species, such as amino acids, nucleobases. So ultimately, we'll try to see whether this particular species, this material, is inherited or not from the very early stages of star formation, to try to provide clues on the origin of life as we know it on Earth. So to try to answer to these questions, we can profit from the synergy between the three what we usually refer to as the main pillars of our field. So observations, usually by means of radio astronomy, laboratory experiments. In the case of, if we target the search for new molecules in the ISM, we usually rely on rotational spectroscopy, and also theoretical computations, either to guide the laboratory searches and also to try to rationalize how the molecules that we see are formed or destroyed in the ISM, thanks to astrochemical modeling. So for those who don't know me, I am essentially a rotational spectroscopist by training. During my PhD, I studied different complex organics in the laboratory. And why do we use rotational spectroscopy? Well, this particular technique can be seen among the most robust technique, if not the most, for the structural elucidation of molecules that first, they need to be in the gas phase, and second, they shall present a permanent dipole moment. But if this is the case, we can think about probably the most simple scenario, so the atomic molecule, for instance, CO. So in principle, this molecule can rotate freely around this rotational axis, and this particular rotation is quantified. So essentially, we can measure the transitions that occur when the molecule changes its rotational level. So if we think about cut in the potential energy surface, and we zoom it only in the ground electronic states, we'll see the different vibrational states here. And then if we zoom it a little bit more, we'll find the rotational level. So essentially what we are observing are energy difference, so changes in this really small gap in energy. But this particular spectroscopy allows us to gain relevant insights into the molecular structure, and also to search for these molecules in space, because the rotational spectra can be essentially seen as a fingerprint of each particular molecular species. So there are different techniques. We can either use the conventional Fourier transform microwave techniques if we are interested in recording the rotational spectra, typically between 2 and 40 gigas, which is usually the first step when we want to characterize the molecule in the laboratory. So we can either use a narrowband technique or a broadband, much more modern broadband Fourier transform microwave spectroscopy. I'll just present the conventional apparatus. So this is a cavity-based spectrometer. This was one of the spectrometers that I used back in Valladolid during my PhD. So essentially what we have here is a vacuum chamber, and inside this vacuum chamber we find two mirrors. So what we generate here is a resonator, what is called the Fabry-Perot resonator. This is coupled with a Fourier transform microwave spectrometer. And also, depending on the particular sample that we want to study, since we need the sample to be in the gas phase, we can either start directly from a gaseous molecule, we can either start with a liquid species and try to heat it, so the molecules are vaporized, or if the molecules are essentially solid samples, we can also profit from laser ablation techniques to vaporize these thermolabile biomolecules directly into the gas phase without any sort of decomposition that can usually occur upon conventional heating. But also, if we are interested in going to much higher frequencies, we can profit from frequency modulation, absorption, millimeter and sub-millimeter wave spectroscopy, and we can reach up to the terahertz. So I'm glad that Lucy Kolesnikova is here in the audience, because she was one of the persons that was involved in the construction of this apparatus. So essentially, here you can see several cells, but in this case, the workflow is relatively simple. We start, the output of a synthesizer is essentially sent into a multiplier change. We multiply the frequency depending on the frequency range that we want to reach. This radiation is sent into the cell, and thanks to a combination of a rooftop mirror and a polarization grid, we double the optical length path of the cell. Then once the radiations come back here, the polarization acts as a mirror, and we collect the radiation back in using a conventional Schottky solid-state detector. So these are the most common techniques that we use in the laboratory. So moving now to the results. The first system, so a couple of systems that I will present, is the study of different glycine isomers. Why glycine? Well, we all know that glycine can be seen as the simplest representative of amino acids, so building blocks of proteins. But despite there have been numerous efforts reported in the literature trying to hunt for glycine in the ISM, this molecule still today remains undetected. So we spotted the need to search for not only glycine, but also other isomers as well as different plausible precursors. And back in 2019, when I was starting my PhD, we performed several theoretical computations trying to form not glycine, but the protonated glycine. In this case, what we found is that we were not observing the formation of glycine, but we found a feasible process under ISM conditions, so essentially a barrierless reaction and an exothermic process leading to this particular isomer when we start with this particular mixture, so acetic acid and protonated hydroxylamine. We were really glad when we saw the detection of hydroxylamine in the ISM that was also reported here at CAP by Victor and Izaskun. And essentially, what we suggested is that the neutral counterpart of this particular molecule, which is this one, acetohydroxamic acid, may be seen as a reasonable candidate for ISM detection. So the problem is that it was quite tricky to characterize this molecule in the lab because it's a hygroscopic solid, quite thermolevel. But using these laser ablation techniques, we are able to characterize both the Z and the E conformations of this molecule. This is what a broadband spectrum typically looks like. So what we do is we perform ab initio or DFT calculations to guide these searches, and these are the typical progressions that we are able to see. So perhaps here you cannot see much. That for us it was quite straightforward to assign different progressions of lines belonging to two conformations, so two rotameric species. But I brought probably a more simple spectra when you can directly see, this is the spectra collected for another molecule for normal butyraldehyde, where you can directly see some progressions that are for which the space is nearly the same. In this case, it was approximately separated by 2 times the C rotational constants. So for us, I mean, it's quite relatively simple to identify these sort of progressions in the lab that we will then guide also the detection in a space. So going back to acetohydroxamic acid, if we zoom in a little bit, in one of these transitions, we can see that each transition can be split into different patterns. And why is this happening? Well, this molecule presents a nitrogen. So rotational spectroscopy is this subtle that it enables to resolve this, what we call the nuclear quadruple hyperfine structure arising from the nitrogen 14 atom, which causes essentially that each rotational transition, it's split it into different components. So this is just one rotational transition. But separated by several megahertz, we observe what we can call a mirror image of this pattern. And what happens here? Well, this molecule also presents a methyl group, and this methyl group can rotate also nearly freely. And what we are seeing is a coupling between the internal rotation of this methyl motion and the overall rotation of the molecule. And what this results is into a splitting of each rotational level into, in this case, 2 substates, which are typically called the E and the A substate. So this is, I mean, this technique is quite subtle. We're able to see how large is the barrier for this particular motion, and of course derive the typical set of spectroscopic constants for these 2 isomers. And with this laboratory data, what we will do, what I will present for the, let's say, forthcoming molecules, is that we prepare a line catalog, and we use this catalog to search for the molecules in the ISM. For this particular case, we search for it toward TMC1 using the Gotham survey, where the molecule was not detected. This is in this slide I'm showing a couple of lines. What we reported was the upper limit to the column density for acetohydroxamic acid. What Brett found is that the upper limit derived for AHA is similar to that derived for glycine, which could be something expected. And we also searched for acetohydroxamic acid toward G+693 back in 2022, this cloud that I've just presented in the introduction. But unfortunately, hydroxylamine was not detected also toward G+693 in this first work. We also reported the upper limit to the detection, and again, it's consistent to the upper limit derived for glycine, so for an isomer of this particular molecule. But then we wanted to keep our efforts on other isomers, and we arrived at glycolamide. So glycolamide is a hydroxy derivative of the well-known interstellar molecule acetamide. So it's a species that, in principle, fits in quite nicely within the chemistry of the ISM. But again, there was lack high resolution rotational data to enable a confident astronomical search. So also during my PhD, I conducted what I call a battery of experiments. So broadband techniques to characterize all the possible arrangement, three-dimensional arrangement, all the possible conformers. cavity-based techniques to fully resolve what is happening? Yes. To fully resolve the hydrogen hyperfine structure. So you can see the difference not only in sensitivity, but also in resolution when you compare the broadband and the narrowband techniques. And also high frequency measurements up to, in this case, almost 500 gigas. So with all this laboratory data, we measured about 1,500 lines for this particular molecule. We again prepare the line catalog. We perform a first unfruitful search for glycolamide towards Cyteris B2N2 in collaboration with the Max Planck Institute for Radioastronomy. But then 3 years later, Victor came to my office and he said, maybe glycolamide is present in the plastic signaling 3. So he re-inspected the data toward this particular Galactic Center cloud. And as you can see here in this plot, well, just for you to know, for the plots that I will show hereafter, essentially we are showing in black the observed spectra overlaid with the LTE model of this particular molecule in red, and also in blue, maybe it's a little bit hard to see, we find the emission of all other molecules that have been found so far toward this particular cloud. So the detection was quite straightforward. It's probably quite interesting that this molecule, as I mentioned, is the first isomer of glycine that has been found in the ISM, and also is the largest molecule and probably the most complex molecule containing an amide functional group in the ISM. And we know that amides are quite important for prebiotic chemistry. In this particular work, we also search for all other possible isomers for which laboratory data, high resolution rotational data is available. None of the other isomers were detected. We reported quite, I would say, accurate constraints on the abundance, for instance, on the abundance of glycine, of both the possible conformers of glycine, glycine conformer 1 and glycine conformer 2. And what is clear is that glycine is less abundant, or it's slightly less abundant than glycolamide. But also this could be a priori expected because glycine has a carboxylic group in its structure. And we know that carboxylic acids are quite elusive in interstellar chemistry, despite they are some of the most widespread species in nature on Earth, and also they are precursors of a lot of relevant biologically relevant species. But besides the detection of formic acid about 50 years ago and the detection of acetic acid reported about 25 years ago, the census of known carboxylic acid detected in the ISM essentially remained untouched. So again, we spotted the need of new and also dedicated observational effort to try to hunt for other acids. And that's how we arrived at this little guy, which is carbonic acid. So carbonic acid plays a relevant role in our planet. It's involved in different biological and geochemical processes. It's thought to be key, for instance, in the anthropogenic acidification of our oceans. Also, it's involved in different processes, so it contributes a lot to the global carbon cycle. And also in space, this molecule has been suggested to be present in different environments, although its presence has not been confirmed in any of them. For instance, some environments such as the Galilean icy moons, in Mercury, North Polar region, perhaps in the surface and even in the atmosphere of Mars, and also, of course, in ice mantles of dust grains, which includes vast amounts of both water and carbon dioxide, which can be seen as a precursor of this particular acid. The problem here is, again, that the laboratory data was a little bit scarce. There were only two beautiful microwave works reported by a Japanese group, by Endo, who characterized the two plausible conformers, so the cis carbonic acid and the cis-trans carbonic acid. In this case, the cis will be extremely elusive for astronomical observations because the dipole moment is super, super low. It's a factor of 15 times lower than that of the cis-trans form. But what we did here is to take this laboratory data, we prepared line catalogs, and we searched for it toward G plus 0.693. We first inspected the low frequency part of our survey, so up to 50 gigas, where we are able to detect directly several lines, such as those that I'm showing you in this plot. But when we started exploring the high frequency data, we started seeing small yet systematic frequency shifts. So essentially what we did here was to take the laboratory data, the previously measured laboratory data that was collected for the first time directly in space. We perform a new global fit, prepare a new catalog and research for the molecule. And this was the final detection that we reported back in 2023. So carbonic acid, cis trans carbonic acid was robustly identified based on different pairs of k equal to 0 lines, rotational transitions with really, really nice signal to noise ratio. So this molecule ranks as the first molecule detected in the ISM with 3 oxygen atoms, and also the third carboxylic acid detected so far. And also it's probably the closest we've been to detect Coca-Cola in space. So just to give some numbers, cis trans carbonic acid, we found that it's a factor of 30 and 7 times less abundant than formic acid and acetic acid, respectively. We also searched for the low energy conformers for cis carbonic acid that, as I mentioned, was not detected. We reported the upper limit to this detection. But based on the upper limit, in case it's close to the real abundance, perhaps carbonic acid is a fairly abundant oxygen-bearing molecule in the ISM. We find that the upper limit of the cis form is in the same order of magnitude as that of the most stable form of formic acid, but it will be essentially invisible to us because of the really low dipole moment. But these sort of studies open us the window to investigate many other high energy isomers. So this task was already initiated here at CAP a couple of years ago, I think back in 2019 by Victor. But in this case, we started exploring not only high energy structural isomers and also high energy Z and E stereoisomers, but we started exploring different high energy conformers. Okay, so because in this case, for carbonic acid, what we are seeing is essentially a rotation around a single bond, but the bond arrangement, it remains exactly the same. So I will just present one example, the one with trans-methylformate, the detection of trans-methylformate, but also very recently we reported the detection of cis N-methylformamide that was led by Sandy Zheng, that is also visiting us this week. So moving to trans-methylformate, in this case, this molecule presents two possible conformers, cis-methylformate and trans-methylformate. The energy separation is about 25 kilojoules per mole, so the high energy isomer is quite high in energy in a, let's say, astrophysical context. There was some laboratory data, although it was, again, a little bit scarce. Again, this molecule has a methyl group, so we will expect this A and E splitting. So the problem is that the laboratory data for the A state stopped at 60 gigas, and the one for the E state stopped at 36 gigas. But there was, with this data, Neil and co-workers in 2012 reported a tentative detection of this particular molecule towards Cytheus B2N, which is the one that I'm showing in this slide, in a detection in absorption. So we wanted to re-explore this molecule. We did this in G plus 7.693, where we are able to, let's say, conclusively report the presence of trans-methylformate, and also we searched for trans-methylformate toward this other region, L1157B1, which can also be seen as a prototypical protostellar shock. So what we did with this study is essentially to confirm that this molecule is present in the ISM, and also we are able to report the cis to trans ratio, so the ratio between the two conformers, which is clear that is far from the thermodynamic ratio. So if we assume that the molecules are the population of both conformers are done according to thermodynamic equilibrium, the ratio will be something like 10 to the 13 to 1. So we will never see the high energy isomer of, for instance, at 100 Kelvin. And also, again, most of these lines have been observed only in space because the laboratory data is sort of scarce. So we have done a recent collaboration with MPE, who have remeasured the laboratory data for this molecule, and we have been able to assign for the first time literally tens of transitions of these high energy isomers. So we need to really take these high energy species into considerations in the context of ISM chemistry. And this is how the detection toward the other source, toward L1157B1, looks like if you account for both the A and E substates. So we know that this molecule is there, but how is the high energy isomer formed? So we ask Germán Molpeceres, a theoretician that collaborates with us, also at the SIC. So one possibility is gas phase routes, starting essentially from methanol, that could lead to transmethylformate. But Germán thought that maybe these high energy isomers are formed selectively directly on the surface of dust grains. So he performed some new gas grain chemical, some surface chemical calculations for this particular reaction. And what he found is that this reaction is sensitive to the stereochemistry. So essentially, he is able to produce these isomers, these conformers in a distinct ratio. And this ratio a priori seems to agree fairly well with the ratio that we found in our data, although the theoretical calculations there great uncertainty. So one take-home message for this is that theoretical computations, also astrochemical models, need to include the whole conformational landscape when we want to deal with the chemistry of these sort of molecules. And now going to the second top of this talk, which is the study of molecules harboring sulfur. As I mentioned, it's a quite hot topic in astrochemistry nowadays. Molecules harboring sulfur are quite relevant because sulfur can be seen as a key element for life. It's present in all major types of biomolecules, so amino acids. It plays a key role in the folding of proteins, also in vitamins and so on. But despite there has been quite recently various detection of many sulfur-bearing molecules in the ISM, its chemistry is not fully constrained. There are several issues. First is that sulfur is not that available compared to other elements, for instance, compared to oxygen or carbon. And also, this element has a tendency in dense environments to deplete quite rapidly onto the surface of dust grains. And this will result in a dramatic reduction of the abundance of sulfur-bearing molecules that we are able to see in the gas phase. And this is what is typically called the missing sulfur problem. I think we've already heard in previous seminars by Asunción Fuente, by Rafa, and many others that are very, very interested in dealing with this issue. So the first molecule that I'll present is the detection of dimethyl sulfide, DMS. So this molecule is the predominant volatile organic sulfur-bearing compound in our oceans, where it's essentially produced by marine biological activity, mainly originated from marine phytoplankton. So until quite recently, it was considered as a unique biomarker in the search for extraterrestrial life. And probably you all have seen these works published by a group from Cambridge. So back in 2023, Madhusadan and co-workers reported what we can say weak evidence of DMS in exoplanet K2-18b, which can be seen as a Haitian world, a planet completely covered by oceans. So as you can see in this plot, this is the James Webb data that they used. So this data is extremely noisy. This work has been highly controversial, and I'm presenting here a collection of works that have already been published refuting the presence of this molecule in this particular exoplanet. But anyhow, well, all this encouraged us to search for DMS in the interstellar medium. The rotational data was already there because this molecule has attracted the attention from many laboratory groups. So we prepared a catalog for this particular molecule. We searched for it toward G+3 and we were able to achieve a first detection of DMS in the ISM. This is just a collection of some of the features that we saw. DMS now ranks as the largest sulfur-bearing molecule detected so far, together with its structural isomer ethyl mercaptan, that was also detected by our group at CAP a couple of years ago. And also, again, to give some numbers, DMS is roughly 1.6 times less abundant than this molecule that I've just mentioned, ethyl mercaptan, and 30 times less abundant than the oxygen analog, dimethyl ether. And even more exciting is the DMS to methanol ratio, that we found that this ratio is extremely close to the value, to the ratio that was found in Comet 67P by Nora Hani and co-workers last year. Of course, in the Comet, this was done by mass spectrometry, so we need to assume that all the mass spectrometric Rosina signal mainly belongs to DMS. But this seems to be the case based on the fragmentation pattern. But if this is true, all this suggests that there might be a chemical link between DMS and methanol. We suggested different possible formation routes. And later on this year, a theoretical group led by Nadia Balokani have studied this particular route starting from methyl mercaptan and protonated methanol that seems to be both feasible and also quite efficient under ISM conditions. So what is clear with all this data is that DMS cannot be considered as a reliable or at least as a unique biomarker in the search for extraterrestrial life in exoplanet science. So the second molecule that I will present is the detection of this particular colorful species, so thionylamide, HNSO. In this case, we use relatively old microwave data by Kirchhoff in 1978. The detection was quite straightforward. We can see a clear detection of a plethora of lines up to, in this case, J equal to 10. So one outcome of this work is that this HNSO, so the most stable isomer of this molecule, is rather abundant. We found that it's only a factor of 5 times less abundant than SO2, which is a well-known, fairly abundant sulfur-bearing molecule in the ISM. And also is the first molecule detected in the ISM containing simultaneously nitrogen, sulfur, and oxygen. So this opens the possibility to explore a link between the chemistry of these elements in a space that essentially remains uncharted so far. So we have started already to investigate some of these molecules. We wanted to characterize the high energy isomer trans HNSO because the difference in energy is not that high and the dipole moment is much more favorable. So we have done this in collaboration with MPE again, using a millimeter wave experiment, in this case, starting with ammonia and SO2. We're able to characterize the FIS HNSO and also to measure in the laboratory for the first time this high energy isomer, trans HNSO. We have just published this work, I think, this Monday, I believe. And last but not least, I will present the detection of oxygen protonated carbonyl sulfide, so HOCS+. In this case, again, we use work by Endo back in 1996, who characterized only the first 3 rotational transitions, so J equal to 0, 1, 1, 2, and 2, 3. So essentially what we did is to treat this molecule as if it was a linear species. We extrapolated this K equal to 0 progressions into higher frequencies, and we are able to detect a plethora of lines up to J equal to 14. So again, a fairly clear detection. This was the fractional abundance with respect to molecular hydrogen that we reported. Again, well, most of these lines have been observed for the first time directly in the observational data. So to confirm all of those, we have performed further laboratory work, again in collaboration with MPE, to confirm all this, that all the lines that we measure belongs to HOCS+, and also to try to hunt for new K larger than 0 lines, that even though this is just a collection of the K equal to 1 transitions, even though they are not clearly detected because they are super weak, the LT model is fully consistent with the observations. So this tells us that the work that we did in 2024 was already quite okay. And also in the first work, we conducted new gas green chemical models to see how this molecule is formed. It seems that HOCS+ is formed through the direct protonation of OCS. Also, this enables us to constrain indirectly the cosmic rate ionization rate of this particular cloud, which seems to be enhanced. This was also seen in the PO+ detection and in the models presented in this previous work by Victor. But also we've presented a complementary way of forming this molecule very recently this year, because we were detecting HOCS+, but in this case, the low energy isomer, so HSCO+, was not detected in the astronomical data. We reported a lower limit isomer ratio, nearly of close to 2. So what we have done is to explore this additional route starting from HCS plus the OH radical, and this particular route in the gas phase seems to be it seems to form both isomers, so HSCO plus and HOCS plus. But if you run kinetic calculations using Mesmer, what you see is that this reaction is preferentially forms the isomer that is protonated on the oxygen end. So this is the branch ratio that we derive. So based on this, it's let's say consistent the fact that we observe only HOCS plus and not the other isomer. And in this final plot, I'm presenting a comparison for the O to S ratio for several pairs of molecules toward different environments. So we see that in black, we are presenting the values reported toward G plus 0.693, which overall are the lowest OS values reported. And in all cases, they are close to the solar ratio that is depicted with this dotted black line. And what we suggest is that this is probably because of the fact that sulfur is not that depleted within this cloud due to the action of large-scale shocks that essentially sputter the ice grains and transfer much of the content that usually in dense environments remains trapped on the surface of dusting and allow us to detect all of these really, really interesting sulfur-bearing molecules. And just a final slide, I wanted to mention the unspoken. So we have reported a couple of beautiful detections, but in parallel to all this work, we have performed the generation of literally hundreds of catalogs of molecules. So we are searching continuously for hundreds of species, and we have been able to identify, to detect a couple of them. So we are quite happy about this. But there are hundreds of molecules that have been searched for, but that still remain undetected. So to conclude, instead of reading some take-home messages, I thought it could be more fun to do a short quiz. So what do you think that causes the presence of DMS in this particular cloud? Option A, aliens, so extraterrestrial aquatic life. Or option B, hands up if you think that this is due to the rich shock chemistry that dominates this cloud. Yes, that's correct, yes. What are some of the main implications of this work? So are we pushing the limits of chemical complexity that is known in the ISM? Does this cloud appears as an astrochemical mine for the detection of new molecules? Space can be a wonderful lab, or do you think that all the above options are correct? Yes, I'm seeing some hands. Yes, that's correct, wonderful. And finally, do these discoveries represent only the tip of the iceberg of what yet remains to be found? Option A, no, we are already done, so I can essentially go home. Or perhaps, but we'll need a completely harmonized theoretical, laboratory, and observational effort. Okay, wonderful. So with this, I would like to thank all the support that I've received throughout this year from all of our group at CAP, and in particular these two persons, so Izaskun for all the help with my very first observations back in 2023, and Victor, you've been a pillar all this year, so thanks a lot.
Thank you, Miguel. Now it's time for questions. So, and thanks for the amazing work, of course, you have presented. Don't be shy. Maybe I can start. So when you talk about the DMS and the methyl ether, you say that the ratio among the two is 30?
Yes.
So what is the difference among this ratio and the elemental ratio, oxygen, sulfur, sulfur-oxygen ratio?
The reason?
This elemental ratio, sulfur.
The element, I mean, I have the value in the very, very first slide. So I don't have it on the top of my mind.
I think it is different, elemental, it's
Yeah,
lower.
yes.
And if this is suggesting some then astrochemical paths that are different between the
between the oxygen chemistry and the sulfur chemistry.
Yes.
I mean, I don't know. The thing is, when we try to see how molecules that have never been studied before in the ISM, when we try to see how these molecules are formed, we usually, in these cases, we usually rely on what has been done before, in this case for the oxygen analog, as a starting point. And in this case, the routes that we are finding are relatively similar. So the routes that form DMS are relatively similar to the routes that form dimethyl ether. The only difference is that in this case we have a sulfur atom. So essentially, if you have in mind, well, we have here one of the possible reactions. So methyl mercaptan plus protonated methanol. We could also have in this case protonated methyl mercaptan plus methanol, and also protonated methyl mercaptan plus methyl mercaptan. But in the case of dimethyl ether, it will be essentially methanol plus protonated methanol. But the route seems to be quite similar. But then regarding the abundance,
yeah, this is why I was asking, because since the ratio is different from the elemental one, if you put this reaction in a model, you would expect the ratio to stay the same. Or there are some rate coefficients that maybe are different, and this is, even if the reactions are the same, this causes then the different efficiency of the different reaction maybe cause the different ratio between the initial one and then the one that you observe.
Okay, but the thing is, the O2S ratio that we are observing is essentially the cosmic, I mean, the solar one in this case, because the cosmic ratio is 31, I believe, the one reported by Asplund and co.
It's the same.
So in this case, it's the same, yes. I mean, it's 30 and 31. So
Okay,
the same,
so it's the same, yes. It's asking.
Thank you for the very nice talk, Miguel. It was really beautiful. I was wondering whether you could comment on the prospects of detecting these cyclic molecules with sulfur in particular in this molecular cloud, given that we had a recent paper on the archive this week.
Yes. So I did not include what Izaskun just mentioned, but we have just published a detection of a relatively large sulfur-bearing ring. So it's a 6-member ring with a sulfur. I don't remember the name of this particular molecule on the top of my head, but yes, a thion. I don't remember right now. Yes. So the thing is, we have found for the first time a relatively large sulfur-bearing cycle, which is quite promising, as you mentioned. So the thing is, the prospects for future detection of related species, I think, are quite promising if we have candidates that have dipole moments that are large enough. So the thing is, in this particular work, we reported the detection of this thion. So essentially, it's a C double bond S. So the dipole moment is quite, let's say, I mean, the bonds are quite polarized. This molecule is quite polar. The dipole moment is roughly 5 device, the mu A. But we are not detecting thiophenol, which would also be expected if we had this other 6-member ring, because both molecules are isomers. But thiophenol has a dipole moment, the mu A is nearly 0.8. So it's less than a factor of 5. So this rationalizes the detection of the one that we see and the non-detection of thiophenol. So I think the path would be to try to hunt for other, perhaps larger cycles. In principle, just with one sulfur, we want to keep things simple, but molecules with fairly significant dipole moments. I think this will be the way.
Thank you. Yeah, 2 questions. So Amelie first and then Antonio.
Hi, thank you for the presentation. I was wondering about the isomers of methyl formate. You said that the abundance ratio that you observe is not the one that you expect from thermal equilibrium conditions. So how do you evaluate this theoretical ratio? Like if the relative stability of the two isomers, it's a ZPE thing, or the 0-point energy difference that you took, or
No, so the thing is, yes, I mean, in this case, the value that I reported is essentially the energy difference between the two conformers, which is, I think it was like 25 kilojoules per mole. So the energy difference is quite large. So essentially what we do is first we assume a Boltzmann distribution at the kinetic temperature of the source. So in this case, we can use, for instance, 100 Kelvin for G plus 0.693, and we see what is the ratio that we get, assuming the delta E derived from theoretical calculations between both isomers. So this includes electronic energy, of course, taking into account the 0-point energy, and we see what is the ratio. And in this case, the ratio was that something like close to 10 to the 13 to 1 at 100 Kelvin. So what is clear is that our ratio can be seen like 100 to 1, something, or 30 to 1. So it's quite far from that expected, assuming this thermodynamic equilibrium.
Okay, thank you.
But this essentially relies on a previous theoretical calculation at the highest level of theory possible to see what is the energy difference. And based on this energy difference, you can directly compute the thermodynamic ratio. It's as simple as that.
Thank you.
Thank you, Miguel, for this beautiful talk. So in your last slide, you suggest that we have to keep on working all together, lab theoreticians and also observers. So this is a very open question, but what's the homework for observers? So what do? We keep integrating to death until the end of times? We switch to different astrophysical environments, or what's the next step?
Yes, my wish list would be to have as many deep surveys as possible. I know that it's really, really difficult to get observing time, but the thing is this detection relies essentially on two surveys, two ultra deep surveys. So the Pepe survey toward TMC1, the Quixote survey, and let's say our survey, our CAP survey for G plus 0.693. And this is what is allowing to detect more molecules. So if we want to, let's say, keep going with the rhythm that we have of detections, we will need to either integrate more on regions that we already know, or go to perhaps other promising regions, but we will need really, really deep data. So my wish list would be to have another regions, perhaps in my particular interest, other shock regions with really, really deep data. But it will take literally hundreds of hours of observing time, so it can be tricky.
Thanks.
So thanks for the great talk, by the way. I thought it was really impressive bringing together these different ideas. So on that point, where are these other shock regions? Because the Sagittarius B2 region in the sort of center of the galaxy is a uniquely rich region in our galaxy that's sort of permeated by these massive stars that ionize the region, which is actually a really great crossover, right? And something I hadn't appreciated because, in the massive star group at the CAB, this is what we work on. This is sort of, the population that we discovered. So yeah, so where are these other shock regions that you think we should aim for?
I mean, there are a couple. One of those is this particular. I mean, this is probably the archetypical shock, I would say. This particular environment, for instance, L1157B1. This is a very, very promising region. And there are some surveys, because in this case, what we did was essentially took publicly available data, the ASI survey of this particular shock. But then there is not that many really, really deep observations of these sources. So these sources, let's say, they are known, but the problem is that we do not have the required sensitivity that enable us to see these molecules. You can see by eye the difference in sensitivity between these two surveys. So even in L with this ASI data that is super, but the sensitivity is still not enough. We are seeing almost at the noise level the transitions arising from methyl formate. So in this case, we did a stacking analysis to improve a little bit the statistical significance, but you can see the difference. I mean, how clear these lines are in G+0.3, and this is essentially a matter of sensitivity, so observing time. So I would say most of the regions are already known, but then you really need to apply for observing time and get really, really deep data.
Okay, thank you. Are there any questions from Zoom? No. So last minute question. Well, you know, Miguel is working here, so if you have any doubt, question, curiosity, you can go to his office. We are at the very end.
At the very end, yes, of the corridor.
Thank you, everyone, for being here. And next week, it will be the last seminar before Christmas.